What's up, Hippie Bio Penguins? So, my students asked me if I could go live tonight for us to do a quick little Unit 3 Q&A review session. So I'm hoping I have enough memory on my phone that I'll be able to save this afterward. But here's to hoping that we're able to do this. We are going to do Unit 3. So Unit 3 is all about cellular energetics. As we've done before, I have my little review screen at the beginning that kind of says what we're going to talk about. So we're going to do something on cellular energetics. We'll talk about cell respiration, photosynthesis, and then of course we'll do a quick little FRQ discussion. And there's 2 FRQs that I found to go over. We have a short FRQ, and then we also have a long FRQ. So let's go ahead and get started. Somebody said, what happens if my class is behind? We're in Unit 2. I honestly do these review sessions also, like my kids are in Unit 3, yes. But I also do these sessions because of the fact that it's a lot easier for you as other students to look at the review stuff when you're about to do it than if I do it after you're already done with it. So my class actually stays ahead of most classes out there. So don't really be worried about the fact that I'm in Unit 3 and you're not. So on to Unit 3. So cellular energetics, we're looking first at the different energy systems that we have, okay? So you can have 2 different types of reactions. You can have an endergonic reaction and an exergonic reaction. So if we're talking about something that is an endergonic reaction, this is an exergonic reaction. What's going to happen with the exergonic is that our reactants have more free energy than our products, which means that we're going to release that free energy. So our free energy is going to be released out. And so you're going to see that we're going to have a negative delta G, okay? That negative delta G means that I have a release of free energy. It also means that it's going to be spontaneous. And of course, we already said that it's going to release energy. Okay, so that's exergonic reaction. Example of an exergonic reaction would be cellular respiration. So in cellular respiration, the glucose molecule has a high amount of potential energy. And as we break down that glucose molecule and we oxidize it, then we're going to release those electrons that have high energy. Then we'll be able to use that energy to synthesize ATP. So the other type of reaction we can have is a endergonic reaction. So in an endergonic reaction, your reactants have less free energy than your products, which means that we had to take energy in from the environment, okay? So this will, of course, have a positive delta G or a positive change in free energy. It has to absorb energy. And then, of course, it's going to be non-spontaneous, okay? So we're going to see there's something called energy coupling. So we're going to have every single reaction that will occur in the world. We're always going to have some type of exergonic that's going to fuel an endergonic reaction. And so ATP and ADP tends to be our energy vehicle that we see inside of our cell. So when we produce ATP from ADP, that's going to be an endergonic reaction. We have to input energy. The energy came from, of course, cellular respiration. And that energy is going to allow us to synthesize or phosphorylate ADP to make ATP. When ATP phosphorylizes something else, it releases that energy. So that would be an exergonic reaction to fuel some other type of endergonic reaction. So let's say pumping something against concentration gradient, moving a motor protein that is carrying a vesicle. This can be any number of things that happen within your cell. And so that would be considered our endergonic reaction. Photosynthesis would also be an endergonic reaction. You have to use some type of energy. So sunlight coming in is going to allow us to then synthesize the G3P or the glucose. There's that quick part. Also in the official Unit 3 is where enzymes are. So let me do a quick little blurb about enzymes. Enzymes' job is to speed up a chemical reaction. The way that it speeds up a chemical reaction is that it's going to decrease the activation energy. And so if you look at my beautiful little diagram, and you can hear my kid in the background. I'm sorry, y'all. So here we see, of course, our reactants. Here's our products. And traditionally, here is the amount of activation energy that we have in order to get the reaction started. Okay. The only thing that an enzyme does is it's going to decrease that activation energy. It does not change our delta G in any way, shape, or form. It still stays the exact same. And it's not going to make an endergonic reaction exergonic or an exer to ender. It maintains the delta G that we have. The only difference is that it decreases that activation energy. It makes it a little bit faster because of the fact that we have less energy needed. Okay. So moving on. So there are 3 main parts to cellular respiration. Okay. Now, and most teachers are going to tell you this, that it's important to know the big picture about cellular respiration. There's a lot of little details that go on. There's a lot of intermediates. There's 10 steps for glycolysis. There's 8 steps in Krebs cycle. There's all these little moving pieces. You need to know big picture. What goes in, what comes out, where does it take place, why is it important? Okay, and so there's 3 main steps, and then there's like this little intermediate steps. You have glycolysis, you have pyruvate oxidation, you have your Krebs cycle, and you have oxidative phosphorylation. So the big thing about glycolysis is it's going to take place in the cytosol. Okay, so because of the fact that it takes place in the cytosol, that tells us that any organism can do it. So theoretically, it's believed that glycolysis was the first kind of metabolic reaction because of the fact that it didn't require membrane-bound organelles, because it evolved before there were membrane-bound organelles, before, of course, mitochondria. It's also going to be anaerobic. It doesn't require any type of oxygen for this step, which is why prokaryotes can do it, and we can do it. That's when fermentation usually takes place, is because we go through glycolysis, and then we have to regenerate a component, and that's why you go through fermentation. So like your muscle cells will go through fermentation because you're going through anaerobic respiration, and you're trying to work out too fast and you don't get enough oxygen. Okay, so of course it could take place before oxygen was found in the environment, and then I think the third part of, there's an FRQ about it. The third part was talking about that it must have basically come through evolution. So since it was favorable in our ancestors, of course, now we have it. So starting material, it starts with glucose, and technically anything can go into cell respiration. You can use all of them, all of the macromolecules except for nucleic acids. And so they'll just enter at different parts, but we go through the parts of cell respiration using glucose as our example. So glucose is what we'll start with. And the end products will be 2 pyruvates, 2 NADHs, and then 2 ATPs. So I always tell my students it's like 2, 2, 2. Just the way to remember it. You probably will not be asked a specific question about how many we make. You'll probably be given a diagram and have to make some type of application, or you'll have to know about that it's occurring in the cytosol and make some type of connection. You're probably not going to have to rote memorize every little part of it. Okay, so that gives us, of course, glycolysis. So ending glycolysis, we have pyruvate. So in order to go into the Krebs cycle, we need to modify our pyruvate slightly. So pyruvate is going to be polar. It needs to move into the mitochondria. So it's going to need some type of transport protein to get in there. So if you don't have that transport protein, of course, you're not going to move into the Krebs cycle, or for the next step for the Krebs cycle. So pyruvate oxidation, which is this intermediate step, is going to take place in the mitochondrial matrix. So there's a kind of region on the inside of the mitochondria, kind of on the inside of those double membrane. That's the inside part is the matrix. And so it's going to start out with pyruvate, and it's going to end up with acetyl-CoA. We're going to release 1 carbon dioxide and then 1 NADH. Okay. Now, considering the fact that we made 2 pyruvates in glycolysis, we would go through this step twice. And so when you're thinking about these questions, they can ask it to you per glucose or pyruvate or per acetyl-CoA or per turn. They can make it per whatever. So you have to follow this up through, right? And so in this case, since we're talking about per glucose, it would actually be 2 carbon dioxides, 2 NADHs, and then 2 acetyl-CoAs. And so that's kind of important that you're following. And the reason why we have 1 carbon dioxide is because you have a 3 carbon pyruvate that becomes a 2 carbon acetyl-CoA. And so since we've lost a carbon, that carbon's lost in the form of carbon dioxide. And this is just a quick little thing that we see. So here is a diagram. You can see the pyruvate. We lose the carbon dioxide. We lose the NADH. And of course, we have our acetyl-CoA. Again, 3 carbons becoming 2 carbons. All we did is add a coenzyme A that helps to facilitate the process. So Krebs cycle is also going to take place in mitochondrial matrix. It starts out with acetyl-CoA. We end up with 2 carbon dioxides, 3 NADHs, 1 ATP, and then 1 FADH2. And so, as we said before, acetyl-CoA has 2 carbons. So it's logical that when I completely break it down, I'm going to have 2 carbon dioxides coming out because I need to completely get rid of all the glucose. So at the end of the Krebs cycle, you have no more glucose present. Glucose is completely gone. We've completely broken it down. We've released all the energy from it, and all that energy is being carried in these different carrier molecules. And so as I said before, you're making 3 NADHs. I always give my students this lie. I tell them that the H stands for holding. That's a lie, but it's okay. So that means that what we're doing is we're holding that electron and we're carrying it to the electron transport chain. They have high energy electrons that's going to fuel the electron transport chain in that process that we're going to take place in just a moment. And so, as we said before, it's important to know per glucose, right? So per glucose, we'll see 6 NADHs, 2 ATPs, 2 FADH2s, okay? Also, I don't think I mentioned it before, but in glycolysis and in Krebs cycle, that ATP is made by something called substrate level phosphorylation. So at the substrate level, we're going to phosphorylate, and that's going to allow us to take that phosphate off to make ATP. We'll be able to phosphorylate the ADP to make the ATP. And so there's that fun stuff. If you look at the diagram though, you're actually going to see that it has a GTP. So I'm sure you definitely know what GTP is, but just in case you don't, ATP is adenosine triphosphate. So it's pretty much just a ribosome that has a adenine on it, and it has 3 phosphates. Did I say ribosome? I'm so sorry. It's a ribose that has the 3 phosphates and the adenine. So it's like an RNA molecule. And then the difference is that a GTP, instead of it being adenine, is now guanine. So it looks the exact same as just a guanine instead of an adenine. So it's a guanosine triphosphate. But just to make life simple, we just say that it's ATP. So that brings us all the way through the Krebs cycle. And of course, when you have questions, you're more than welcome to pop them down in the little question box. Oxidative phosphorylation has 2 steps, okay? So this is going to take place in the mitochondrial cristae. So basically that inner mitochondrial membrane is where we're seeing this. 2 steps. The first one is going to be electron transport chain. The second one is going to be oxidative phosphorylation. And so with the electron transport chain, what we're doing is we are transporting electrons. So the electrons have this high energy. They're in NADH and FADH2. They're going to bring it to the electron transport chain and donate those high energy electrons. As those electrons fall down in energy, so as we start here and they fall in energy, they're going to pump protons across the membrane. So there's all these protons being pumped into the intermembrane space. It's important to know where it's being pumped. It's being pumped into the intermembrane space. And so we're going to see that there's going to be a high concentration of protons in this area. Okay, that is all you see with the electron transport chain. There's no ATP made. All we're doing is pumping protons across the membrane. A second part was chemiosmosis. In chemiosmosis, we're going to use this enzyme called ATP synthase. Those protons are going to fall down their concentration gradient through ATP synthase, which is kind of like a little motor, and it rotates. Okay, so as it rotates, it's going to phosphorylate ADP to make ATP. Okay, and so in the second part, that chemiosmosis is where we make the ATP. So input would, of course, be our electrons from NADH and FADH2, and then it's going to, the out product is going to be our, of course, ATP. They're not going to ask you the specific number, but there is more ATPs that will be made in oxidative phosphorylation than the other 2 steps. So we saw in glycolysis there was 2, Krebs cycle there was 2, and then we have approximately like 36-ish in oxidative phosphorylation. So there's a lot more ATP that's happening here. And so if there's any type of process that inhibits NADH from releasing its electron, that's going to back up this pathway. If we don't have any oxygen, it's going to back up this pathway. And so you'll see that there could be questions asking if Krebs cycle doesn't occur, if the ETC doesn't happen, what will happen to the pathway? And so you have to make sure that you can connect all the dots to realize that NADH is what fuels this. If you can't release your electrons, you can't go through Krebs cycle, nor can you go through electron transport chain, and you're going to make less ATP. So that's a big thing to keep in mind when you're thinking through all these processes. So moving into photosynthesis. So photosynthesis has 2 main steps. There's the light reactions and there's the Calvin cycle. Okay, so I didn't mention this before, but cell respiration takes place in mitochondria. All organisms have a mitochondria. Okay, so our plant cells, they have a chloroplast that goes through photosynthesis. So although they're going to go through this process to make their own food, they still have to break down that food using their mitochondria. All the ATP that we see in photosynthesis is going to be used in photosynthesis. That ATP is not used for cellular energy. All cellular energy is going to come from cellular respiration. So as I said before, there are 2 steps. We have the light reactions and the Calvin cycle. So the light reactions will take place in the thylakoid membrane. So there's these little stacks that you see inside your chloroplast. This little stack is called a grana, and that membrane is your thylakoid membrane, okay? You're going to start out with water and sunlight, and then it ends up with ATP and NADPH. Yes, notice there is a P there. When we were talking about cell respiration, we said NADH. Now in photosynthesis, we have NADPH, okay? A reader on an AP exam is not going to know that you meant to do the other one. Okay, so be very, very careful when you're writing these words that you're using the right term. P is always going to be in photosynthesis. NADPH is only photosynthesis, while NADH will be cellular respiration. Okay, so in our light reactions, the big thing that's happening here is that the light is shining down, right? There's two main photosystems. You have photosystem 1 and photosystem 2. Now, because of the fact that they're named based on when they were found, we actually see that the energy goes through photosystem 2 first. Then it will go through photosystem 1. And so let me show you our little graphic to make life just so much easier for us. So light kind of shines down here on the pigments. So inside of this photosystem, PS stands for photosystem. Inside of the photosystem are all these pigments. The pigments are going to absorb that light energy, and it causes their electrons to go to a higher energy state. When that happens, there's this electron acceptor up here that's going to seal the electron, and it goes down this electron transport chain. We saw electron transport chain again. So as the electron falls down in energy, it pumps protons across the membrane. Okay. Now, it's important to realize it's being pumped into the thylakoid. Inside that thylakoid membrane is where we're going to pump our protons. Okay. So that's slightly different than what we saw in cellular respiration, and that difference is important to know. So the electron goes down the electron transport chain, allowing us to make our proton gradient, which will later on allow us to make ATP. That electron is going to end up going into photosystem 1. Light shines down, which of course causes the electron to jump to a higher energy state, and it goes down the electron transport chain to go into NADPH to hold that electron. And as I said before, we do have an ATP synthase that's going to allow us to synthesize ATP. So when we look here, this is considered linear electron flow. We start here in photosystem 2. We go through an electron transport chain making ATP. We then have another photosystem, and then we go through the process of making NADPH. Okay, so we're making a 1-to-1 ratio between ATP and NADPH. This is important. Energy went straight through. Okay, the electron went straight through. Okay, and so it's important to realize how we're able to make that. There's another type of electron flow you can have. This is called cyclic electron flow. With cyclic electron flow, all we deal with is photosystem 1 and this electron transport chain. So the energy, the light energy, shines on our pigment, causing it to jump to a higher energy state, and it's going to keep cycling through this electron transport chain. By doing that, it's going to keep making that proton gradient, and we're going to end up being able to make more ATP than we make NADPH. This is important because in the Calvin cycle, we need more ATP. You're going to need 9 molecules of ATP, but only 6 molecules of NADPH. If we only had a 1-to-1 ratio, it's going to have that same buildup that we mentioned before with cellular respiration, and it's going to stop the process from taking place. And so students have asked in the past, well, how do I know if it's going to be linear, electric, or cyclic? And when the photosystems and everything is organized, it's actually made where this specific section is linear, this specific section is cyclic, and I'm assuming genetically it just makes them work out the way they need to be, just evolutionary it's worked. So the second part we have is the Calvin cycle. So the Calvin cycle is going to take place in the stroma, okay? So it's like the cytosol of our chloroplast. We're going to start out with carbon dioxide, the ATP that we made in the light reactions, and the NADPH that we made in the light reactions, and we'll end up making something called G3P. So there's 3 main steps that we have in our Calvin cycle, okay? So we bring in 3 carbon dioxides, and they bind with something called RUBP, ribosylose bisphosphate, okay? Not important to know the name, don't worry, y'all. So these come together making some big, long compound, okay? This goes through this process where that carbon dioxide comes in. It's called carbon fixation. It then goes through a reduction phase. In case you don't know, reduction is gaining of electrons, right? So NADPH has the electrons. So it's going to donate those electrons to this pathway, right? It's going to donate those electrons to that carbon-containing molecule. And so since it gains those electrons, so this is called reduction. And so NADPH is going to, of course, gain that electron. NADPH is going to donate that electron as well as this ATP in order to make that G3P. One comes out, and the other 5 will then go through the last step, which is rearrangement, to rearrange in order to make the RUBP again to allow for another part of the cycle. Okay, so my phone is dying soon. So we're going to rush through these FRQs, and I'm going to pray that I can hopefully have enough battery to get through, as well as have enough memory on my phone to be able to save this video for later. FRQ question that we're looking at is 2019 number 3, in which we have this big background. It's a short FRQ. It says pyruvate dehydrogenase complex, PDC, catalyzes the conversion of pyruvate to acetyl-CoA, a substrate in the Krebs cycle. So they tell us this is something in the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency rare. So we first need to come up with where's the cellular location where the PDC is most active. Well, since we know this is happening with the Krebs cycle, I know this must be taking place in the mitochondria, specifically in the mitochondrial matrix. Okay, so that would be our answer we should see here. You can either just go straight with mitochondria or go mitochondrial matrix. If you tell me the Christe, you are wrong and you will not get this point. So you do need to make sure that you understand the location of where we see that Krebs cycle taking place. So the next part says, make a claim about how the deficiency affects the NADH produced by glycolysis and the NADH produced by the Krebs cycle. Since it specifically tells us this happens from the pyruvate to acetyl-CoA, glycolysis makes pyruvate. So logically, the disorder isn't going to affect glycolysis and the NADH that's made in glycolysis, because this disorder is affecting something that's happening after glycolysis. And so you would see a no change for glycolysis because of the fact that glycolysis occurred first. And then you would see a decrease in the Krebs cycle because of the fact that this pathway is going to take place and it's going to slow down, right? So we're going to see this greatly reduce, slow down, since there will be a less acetyl-CoA. Since you have no acetyl-CoA to go into the Krebs cycle, you will see a decrease in this process, okay? And the last part is looking at genetics, and my students haven't learned about the genetics yet, so logically this can't be a question that they see tomorrow. But what can see has happened is that X chromosome. So males, just so you're clear, in biology, we talk about males being XY and females being XX. I know there are many other genders out in the world. And so we're just talking about like assigned at birth, right? So male assigned at birth has an X and a Y. Male assigned at birth has XX. And so what we see here is that they're saying that a male with PDC deficiency and a homozygous female with no family history have a male offspring. Calculate the probability male offspring will have the PDC deficiency, okay? Well, if the male has it, he's going to pass on his Y chromosome, right? So here's our male affected. The only thing that he can pass on to his children is that Y chromosome. So there's no way that that trait can be passed on, so there'd be a probability of 0, okay? The offspring will not and cannot have the PDC, okay? And so that is our short FRQ. Our long FRQ that we can have is to help ease your minds, to show you, hey, there's all this stuff that we want in cell respiration. Make sure we understand big picture, right? And so here they give you the glycolysis and pyruvate oxidation. Here they're showing you what the Krebs cycle looks like, and here's the electron transport chain. So cellular respiration includes the metabolic pathways of glycolysis, Krebs cycle, electron transport chain as represented in the figures. Cellular respiration, carbohydrates and other metabolites are oxidized, aka lose their electrons, and resulting in energy transfer reactions to support the synthesis, okay? So they're wanting us to say, how do each of these steps contribute to ATP synthesis? When we look at this picture, we can see, well, look, we make ATP. So the synthesis of ATP by substrate level phosphorylation. Also, you make pyruvate and acetyl-CoA. That acetyl-CoA is then going to move into the Krebs cycle to continue the breakdown so we can synthesize ATP. We also make NADH. NADH is going to go to the electron transport chain in order to create that proton gradient so that we can obtain that ATP. So all of those options are available here as different answers you could have put. NADH and ATC, acetyl-CoA for Krebs cycle, as well as energy for substrate level phosphorylation. Okay. And yes, I'm speaking very quickly because I'm trying to make sure that I get done before my battery runs out, right? Plus you can hear my kid crying in the ground, which means I need to go help my husband with the baby. The second part, looking at intermediates of the Krebs cycle. Okay, so if we look at our Krebs cycle, again, we make GTP. GTP can be used for substrate level phosphorylation, and we can make ATP. We also have NADH and FADH2. That's going to allow us to move to the electron transport chain in order to make ATP. Okay, so all of those are definitely things you could have producing NADH and FADH2 for ETC, releasing high energy electrons for the electron transport chain, providing the energy to pump protons for that concentration gradient we see in the ETC, and then of course looking looking at GTP for our substrate level phosphorylation to making ATP through ADP. And then the last part, looking at, of course, how does our proton gradient allow us to make ATP? So NADH and FADH2 are dropping off their electrons, pumping those protons across the gradient. That the proton gradient is then going to move through ATP synthase in order to synthesize ATP. So this is like a potential energy. And so we have this stored energy in that proton gradient that allow us to move ATP synthase in order to synthesize ATP. Whew, mouthful, right? So part B is looking at evolutionarily, right? So why can we say that glycolysis occurred in a common ancestor? Well, we already went through this a second ago, right? So it tells you that nearly all organisms perform glycolysis. It occurs in anaerobic conditions, and then it also only occurs in cytosol. So if you look at this, you can say, okay, well, since we're saying that it occurs in all organisms, it must be due to natural selection. An organism out there was more likely to survive because it was able to make this ATP. And since it was able to survive, it passed down that trait. So this trait has been inherited or passed down throughout generation to generation. The second part, looking at anaerobic conditions. Well, it must have occurred and originated before atmospheric oxygen. I think we all know that photosynthesis is what generated that oxygen in the environment. Oxygen again started in the water, and once it built up the highest concentration, it then was released out into the air, and that's why we have so much more atmospheric oxygen now, is because you can't keep all that gas in the water. So the last part, I was looking at glycolysis only occurs in cytosol. Well, that must mean that it predated the membrane-bound organelles, so it predated any type of mitochondria or other organelles that would be taking place in the cell, right? Which is where we see the last two steps of cellular respiration. Whew. We also had to do a calculation, okay? So it told you about the amount of energy you have in glucose. You have 686 kilocalories per mole. It also tells you that converting ATP to ADP, that we have 7.3 kilocalories per mole. So you have to calculate the number of energy, or how many moles of energy you have when you have 30 moles of ATP. So if you just take 30 times 7.3, because this is how much you have for ATP, gives you 219 kilocalories. Well, now we have to figure out, well, how efficient is that? So I just take that glucose is 686, and I do 219 divided by 686, giving me 0.319. So it's either 31 to 32% efficient. And then it asks us, well, where does that extra energy go? This 400 and whatever that we're missing, where does it go? Well, of course, all energy is going to be released in the form of heat. And with that, I have finished. So here are a bunch of game codes in case you are not one of my students. Here's a bunch of game codes, 2 different quizzes games. In case any of those don't work, you could always just check out the Weebly because all the codes will always be up to date on there for you. And so with that, hopefully my phone has enough juice and that there's enough space for me to save this so that we'll be able to save this for the future. I hope everybody has learned something. Remember your AP Bio Penguins and your dress for success. Good night and good luck tomorrow on your test. Bye, y'all.